want to isolate that plasmid you put in, then you can do this by taking things for a mini prep spin. So Miniprep, or alkaline lysis, is a way that you can purify out a plasmid that you put in bacteria. And so these plasmids are circular pieces of DNA, and we can get bacteria to make lots and lots of copies of them, either because you want the DNA or because you want to get the bacteria to make protein from them. So when you want to have lots of copies of the DNA, you want to make sure that the DNA is pure so you can send it for sequencing. And you just want to have that plasmid so you can keep a stock of it, various things. You can use it for PCR, so there's lots of reasons why you would want to get that plasmid out of bacteria after you get the bacteria to make a lot of copies of it. So now we can purify it out using this mini prep kit. And so here's how it works, because knowing what's going on in each of these steps is going to help you if you need to troubleshoot, and it's also just really cool. And it's always good to know, even when things come in the box, what it's actually inside the box. So the kits look something like this. You don't have to use a kit. I will give some links to some make-it-yourself versions of these. And in fact, when we do like baculovirus purification, we often don't do it with a kit. We just make the individual components. Okay. But so what is in these components? So the basic idea of what's actually going to happen is that we're going to take that like 5 mil or so culture that we grew overnight, and we're going to spin it down. And when we spin it down in the centrifuge, this is going to pellet all the cells. And then we can pour off the media into some like bleach solution, or we can bleach it afterwards. Then what we want to do is we want to resuspend it into better conditions. And so it was in this bacterial media, and that bacterial media has all sorts of stuff that you wouldn't want. It potentially has like DNA chewers and RNA chewers. We don't care about the RNA chewers, but the DNA chewers would be a big problem. Other stuff we're trying to purify basically. And so we want to make it as pure as possible in an environment when we break open the cells, which is going to be the lysis step. But first we resuspend it in this clean buffer. So we take this, we spin it down in like the culture tubes, then you just resuspend it in the tube. And now you're going to add it to a clean tube. So this is not, you're not going on the column yet. First, we're going to precipitate things out. If we just put it on the column now, it would just clump up the column. Instead, what we want to do is we want to remove all the insoluble stuff. So right now we've removed the media, but if you think about a bacterial cell that's got that whole membrane, it's got all sorts of proteins, it's got tons of stuff other than the DNA. So we want to get as much of that removed as possible before we go on to these purification columns. So what we can do is we can lyse it, we break them open, and I'll show you how we do this with detergent. So this detergent called SDS. It's going to work like soaps work. As a detergent, it's going to break up the bacterial membrane. And then you have NaOH, so you have sodium hydroxide. The space this is where you get the alkaline. In the alkaline lysis, this is going to basically, it's going to denature the DNA. It's going to make the strands come apart. Now, when those strands come apart in your plasmid, it's okay because your plasmid is pretty small. It's all like super coiled, so it's all coiled up together. So if you make the strands come apart temporarily, yeah, it won't like you that much, but it'll come back together quite easily. But you take this bacterial genome that's also in there, you have all this giant bacterial DNA, which we don't care about. We want to get rid of. That's not the DNA we want to stick. So we want to get rid of it. And how we can do this is because it's so big, when it comes apart, when the two strands come apart, they're going to have a much harder time finding one another. And so this is going to make it like precipitate out. And so it's going to come out in the insoluble stuff. So now we're still before we're column. First we're going to neutralize things, because right now we're at a really basic pH and we don't want to keep it at a basic pH. So we're going to neutralize this so that the DNA strands can come back together if they can. So your plasmid strands can, but the genomic DNA, the gDNA can't. And so you add potassium acetate in your neutralization buffer. This is going to lower the pH back to allow the strands to zip back together. And then there's potassium. So the potassium part of this, so the acetates go in the pH. The potassium is going to precipitate out the mess that it's tangled in. We'll get into this more, but so potassium didecyl sulfate is not soluble. So SDS, sodium didecyl sulfate, is soluble, which is why we can use it to when we're doing like an SDS page and we want to denature our proteins. But if you have ever done an SDS page and you've tried to run your sample and it's clogged up the well because you have KCl in your buffer, that's because the KCl, the potassium, is going to precipitate out the SDS. And so this is going to, because the SDS, the decyl sulfate part's like bound to protein, it's bound to all the stuff. Then when you have the potassium, that's going to precipitate it all out. And so this is going to allow you, when you spin that down, you're going to get this like whitish, clumpy stuff on the side of your tube. And then you're going to remove the supernatant, so the liquid that's left over. And then you can use this column. And when you put this on the column, and this in beginning, you have it in this waste receptacle thing. And then later we'll do the washes, and then we will elute it. So get it off the column and into a nice clean tube. So let's go with a little more detail in each of the steps. So we've taken our colony, we've grown it up overnight, depending on how many samples you have, or if it was a cloning, and so you're trying to test to see if a lot of them have it, or if like you're going to send a bunch for sequencing, or if you just need one copy. Basically, you can have a different numbers of tubes depending on what you're doing. Sometimes you can have a lot. In that case, they have like vacuum manifolds where you can do the steps in a vacuum as opposed to doing them in the centrifuge, which is the method that I normally use, and I'll show you. But if you are doing it in a centrifuge, make sure that you don't do more than like 24 or 12, whatever your centrifuge can hold at a time. Okay, so now we have done that. We've taken our growth and now we spin it down, and we're going to do the resuspension. So what's in this resuspension buffer, this buffer P1? The buffer P1 is normally kept in the fridge. It'll come in a box. And then what you do is there's an RNase that you add. And you add this, and then you keep it in the fridge. So this RNase A is going to degrade the RNA once we open the cells, but the DNA is going to be safe because it doesn't have the 2' OH. And so as I talked about in my post on my RNase is so dangerous when you're working with RNA is that RNA, it has this 2' OH, and RNAs can take advantage of that 2' OH and use it to make the RNA attack itself. And so RNase A is this enzyme though. And so if you've worked with enzymes, you know, you don't want to like keep them at room temp and stuff. They're not going to be happy. And so you can, when you stick it in this buffer and resuspend it, then it can do. So it'll come in like a separate little vial, and then you add this and keep this in the fridge. Okay, and then but your DNA is going to be okay. It also has, but there's a little box on the top of the bottle that you check off once you've added the RNase A, so people know that it's been added when they go to use it. Okay, what else is there? There's the buffer part. So a buffer pH stabilizer. So it's got like an acid part and a base part form, and it goes back and forth, donating a proton if it needs to lower the pH, make things more acidic, or take a proton if the pH gets too low, too much proteins around, then it gets happy and takes it back. And so this is going to help stabilize the pH. And at this point, we're keeping it around 8. What else is in here? Oh, and so we also know that thymophylline is going to serve as a pH indicator. So this is like the lyse blue stuff. And so this is another thing that you can add to the buffer. You don't have to add. This is going to serve as a pH indicator. So later it's going to turn blue when we get to high pH. And then later when we neutralize it, it's going to go back to colorless. And this is going to help us see that our things are thoroughly mixed, that our pH is all good and everything. Finally, this has glycerol. This is going to keep the cells from bursting for now. It's going to act as like an osmotic balancer. So osmosis is when you have like the water or some other solvent moving from an area where there's not much solute, so not much stuff dissolved, to an area where there is a lot of stuff dissolved. And so if you think about our bacterial cells, there's a ton of stuff in there, not much stuff out of there. And so if we didn't add something like glycerol, it's just going to kind of provide a bunch of bulk, but not really mess with anything, then everything would burst because all the water would go into the cells, and we're not ready to burst them yet. We just want to get them in the nice environment so that we can burst them in the lysis step. So what's going to happen in this lysis step? So first of all, what we're facing is the bacterial cell is surrounded by these like membranes. to lyse it, we need to destabilize that membrane. And then we're also at the same time, we're going to disrupt the DNA. So this is where to get the membrane destabilized, basically this membrane is stabilized. It's like has these negatively charged phospholipids and polysaccharides, LPS, and then it has this magnesium and calcium that are going to help stabilize and neutralize those charges. So these are positively charged, they're going to bind to the negatively charged membrane and help neutralize them. And this is how the bacteria normally keeps its membrane happy. But if we add EDTA, EDTA is this chelator, so basically it can clamp on around metals and hide them from the bacteria. And so when it takes that away, now you're going to have all those negatively charged phosphate head groups that are kind of like trying to get away from one another. And then you have this dodecyl sulfate from your SDS that's going to kind of sneak in there now because it has this long hydrophobic region. It's going to sneak in. So basically the membrane is going to get disrupted. And now we're going to take advantage of the pH. We're going to selectively, keep our plasmid DNA while getting rid of the genomic DNA when we spin it down and make it precipitate out. And another thing is that the SDS is going to denature the proteins that are in this membrane, because normally we like picture it and stuff as we think about it as this big, like all this lipids, but there's really a bunch of protein in there. Okay. And so now the DNA. So the DNA strands are held together by these hydrogen bonding. And so hydrogen bonding must form another post, but basically you have this hydrogen and it's partly positive because it's attached to something electronegative that's going to be pulling these shared electrons. And then you have an acceptor that is going to be something that is electronegative. So something that's really pulling on electrons, and it has like a lone pair. So it's just an oxygen or a nitrogen. So, and you have this like attractions, these partial charge attractions between the strands of the DNA that are helping keep them zipped together. So this isn't the bonds that are holding together the individual letters in a strand, but rather between strands. And so we can disrupt this by disrupting the hydrogen availability. So hydrogen bonding, take away the hydrogen, you can't get the hydrogen bonding. And one way you could take away the hydrogen is by going to an alkaline pH where there is fewer protons available. And so by going to this low pH, you're able to disrupt the strands. And note that you can do this for the DNA, but you can't, if you were to do this with the RNA, it degrades at the hydrolytic, by this like hydrolytic cleavage at basic or alkaline conditions. So basically it's doing what RNase A made it do before, but here it's doing it just because the pH is low, not because RNase A is helping pull off that proton. Okay, so we have unzipped our DNA, and now it's time to rezip our DNA. And remember that the big genomic DNA, it's too big, it can't find itself easily. It's going to get tangled up in this whole big SDS coded mess. And then the KDS is going to basically, now you have potassium in here. The potassium is going to desolubilize this because it's going to precipitate out with the DS. And then you have your acetic acid that's going to help neutralize the pH so that those strands can get protonated again and they can come back together. And then you're going to have guanidinium chloride, which is going to help it bind to the column in the purification step. And so this purification step is going to happen in one of these little columns. There are ways that you can do things without a column. So these are the columns that come with like this Miniprep kit, and you can see that they have basically like a capless weights tube. And then you're also going to need clean cap tubes. So before we had a clean cap tube when we were doing the lysis, the resuspension. Now this is a really clean tube. This is a different tube. But first we're going to do all the washes and stuff into this waste tube, and then you transfer it into the clean tube. So you can do it in the spin method, or you can do it using like a vacuum manifold. The point of the centrifuge and the point of the vacuum manifold is to pull things through the column rather than trying to wait for gravity to pull things through. So speaking of the column, basically it's going to be like a silicon column, and you're going to, it's very similar to if you've done like a PCR purification kit. So basically you get the conditions right for the DNA to bind. So we're going to have a chiotropic salt. It's going to like break up the water coat, and then We're going to help make the DNA find and bind to the silica gel membrane. And then the pH is really important, especially when you're doing like a PCR purification. Often you have to like check the pH and make sure it's okay. Then you're going to bind it on there. Use wash with an ethanol wash. It's going to get rid of the salt, but keep the DNA on. Then you spin it again to make sure you get all the ethanol out. Then you can raise the pH again so that you deprotonate the membrane. So they're going to bind at an acidic pH, so a low pH. Basically, the silica in this membrane is going to then be protonated, so it's going to be neutralized. And this is going to allow the DNA to bind because remember, DNA has a negatively charged backbone. And so if this was negatively charged, DNA wouldn't bind. Then you have a high salt and you have a guanidinium. This is a chiotrope. So basically what it means is it's going to like loosen up the water coat surrounding the DNA. It's also denaturing any of the proteins and helping shield the DNA's negative charge so that the DNA and the silica can get close and form this more extensive bonding. Because DNA has that negatively charged backbone, this guanidinium is going to help shield the backbone so that it can get close to the silica. Okay, and then you have like isopropanol, which is going to help them find each other and kick off the water. Okay, so basically, yeah, that's going to allow you to now wash the stuff. And so when you do you do like a buffer wash. So this buffer PB. It's just going to wash things, and then you add the PE buffer. So this one you're going to have to add ethanol to. If it hasn't been added on the top, there'll be a little thing like a box like we saw with the RNase A or with the P1. But here it's for the adding the ethanol because you add it. It doesn't come with the kit. So you add the ethanol. The ethanol, what it's going to do is it's going to get rid of the salt. It's going to solubilize the salt, but not the DNA. So then you can wash the salt away. But it's also going to interfere later. So what you want to do is you spin it without adding any more ethanol. It's going to, without adding anything more, it's going to pull down through any remaining ethanol that didn't get through in your first spin. Okay, now you can elute. So it comes typically with like an elution buffer, but typically you just do it in pure water. So one of those waters that you actually buy that says like RNase, DNA is free. And this water, now you're going to have lower salt and higher pH. So all the things that made the binding buffer really good for binding, you're now going to reverse those things. And so your DNA is going to repel from the membrane and your DNA will come out. And then you can check the purity and concentration with something like a NanoDrop. So hope that helped.